Okay, good afternoon, everyone. Hope things are warming up for you wherever you are. Today, we're going to do the first in this year's series of Child and Adolescent Division sponsored Grand Rounds, and are very excited again to have Dr. Joan Luby with us from St. Louis. And God willing, her presentation this time will not be marred by technological problems. Dr. Luby is the Samuel and May Ludwig Professor of Psychiatry at the Washington University School of Medicine, where she has founded and leads the Early Emotional Development Program. Dr. Luby has specialized in infant and preschool psychiatry, and her program of research has focused on early childhood psychopathology and emotional development, specifically in application to the risk trajectory for early onset depressive disorders for almost 30 years. She has conducted some of the first large-scale empirical studies of preschool onset depression and provided data on the validity, clinical characteristics, longitudinal course, and brain developmental outcomes of early onset depression. And with her colleague, Dr. Deanna Barch, they are in the third cycle of development in a study sample enriched for depressive symptoms arising as early as age 3. Dr. Luby's studies have established the powerful role of early childhood caregiver support and psychosocial adversity on brain development, as well as the mediating role of maternal non-support on alterations in brain and behavioral outcomes related to early exposure to poverty. And more recently, Dr. Luby has developed a novel early parent-child intervention for depression that focuses on enhancing emotion development, and this has showed strong effects and neural change. So without ado, and with many thanks again, I give you Dr. Joan Luby.
Thank you, and thanks for coming today. Hopefully everyone has this text code, so I will just move on from there. So today what I'm going to focus on is how early experience sculpts brain development and how we can use this information to better optimize neurodevelopment in early childhood as kind of a public health agenda. This is just my conflict slide. So fundamentally, I'm going to talk about a formula for growing healthy humans, essentially developmentally informed health promotion. And this formula capitalizes on the principle of experience changing and sculpting neural systems, and particularly powerful during early sensitive periods of neurodevelopment, which is happening in the first 5 years of life. So this research focus is something we kind of stumbled upon. As Dr. Sauls just mentioned, we've been following a study sample of a little over 300 children from the St. Louis metropolitan area. We captured them when they were between the ages of about 3 and 5. We oversampled children who had symptoms of depression, and we began following them over time, assessing their behaviors, observing their interactions with their caregivers, assessing the primary caregiving relationship. And then when these kids became school age, I began a collaboration with Deanna Barch, who is a neuroscientist, and we began doing brain scans on these children. And right now, this study sample is edging into early adulthood, and we have 5 waves of brain scans on the sample. So we've been able to take a look at how early childhood depression impacts the trajectory of brain change. But what we also stumbled upon were very, very powerful effects of other aspects of the environment on brain development, in particular, poverty and adversity, and maternal support. So these are things, we basically found these very, very strong effects of socioeconomic status on brain change. And we first even included that because we were sort of controlling for that in our analyses, but we found they had these huge effects. So we began to start to chase that down and become very interested in that. We also found that these effects were particularly powerful during sensitive periods, and I'll tell you a little bit more about that later in the talk. We also found that there was some regional specificity of the effects of experience on brain development. And I do think this is interesting news and probably validating to a community like your own, where you've been very, very committed, at least historically, to the practice of psychotherapy. And I think what this sort of underscores is the importance of these experiential factors on brain functioning. Okay, I'm trying to advance. Let me see, here we go. Okay, so some of the key principles that we drew from this are that experience materially impacts brain development. And we know this because there are strong associations between experience and brain development that have been demonstrated in multiple longitudinal studies of humans. We know that this is causal because people who have studied animal models and who experimentally change the environment and look at how these changes impact brain, we know that these are causal. Obviously, we can't do those types of causal studies in humans, but we know we can infer causality. We know that there's causality from animal studies. What we can do in human studies, and this is kind of what I'm arguing should be done going forward, are you can do enhancement studies. You can take developing children and you can sort of randomize children to enhancement conditions where you teach them certain types of skills more than occur in standard daycare, preschool, or care, and you could see how this changes both brain and behavior. We know from a whole body of literature that the impact of experience on the brain is particularly powerful during sensitive periods. People have utilized these sensitive periods when we think about learning how to play the violin, for example, or we think about learning languages. We know that there's certain times in development when this learning is much easier and faster, and the brain is essentially more sponge-like to take in experiential stimuli or to learn. So what I'm arguing is that these principles, the effects of experience on brain development and the fact that they're powerful during sensitive periods, should be used to inform public health planning for optimizing child neurodevelopment. So if we think about health promotion in primary care, if we, children who go to see their pediatrician, immunization schedules, for example, are determined based mostly on animal models. And things like how we feed children and what we feed them is really not that detailed or scientific. I'll talk a little bit more about why this is important in terms of this whole new area about the gut microbiome and its importance in health and its importance in brain development, which suggests that diet and also the psychosocial environment very much influence the gut microbiome. We know the gut microbiome is very, very important in health outcomes. We're learning more about how important it is in brain outcomes. So new data is available that informs the timing of exposures to antigens and allergens to reduce allergies. This is sort of a very, very new area in the study of allergies, that we can actually expose children to certain types of allergens early in development. And this has shown a lot of promise to take children who have severe allergies and minimize, to sort of coach the immune system not to respond to these allergens. Another really important aspect in brain development that we have yet to really seize on in sort of primary care and public health models are sleep. We know that sleep has very, very important effects on brain development. Sleep is very restorative. There's certain aspects, certain phases of sleep during which there is sort of neural cleansing taking place. And we know that this is also particularly important early in childhood. But we need to know more about the details of sleep, what phase of sleep is important, when it's important in early life, to inform how it can best enhance brain development. So all of these things related to the timing and the nature and how these things work, which are mostly now just sort of hearsay or opinion or based on sort of standard practices that aren't necessarily empirically informed, could be very much more informed by data that we have about how these experiences impact the brain and impact the immune system. And I'm talking about the immune system because first of all, the immune system relates to the brain, which I'll tell you a little bit about later, but also because the immune system is the consummate model that is informed by experience. Our immune system is designed to respond to experience, to figure out what is toxic, what is not, and how to mount a response or a non-response to environmental stimuli. So what I'm arguing for is that we can have precision early developmental health promotion and enhancement. We don't currently have that in the country or in the world. And there's been surprisingly little attention to key methods to enhance this early in life. It can help in our understanding of risk trajectories and sensitive periods. And these are most important for children to be protected from risk and when they can most benefit from exposure to stimuli. So if you think about it, people who grow vegetables are particularly, if you think about big industries like Monsanto, where they're growing, it's now not called Monsanto anymore, I'm dating myself, but since it's in Missouri, I'm sort of acutely aware of it, that when these big corporate industries are trying to sort of gain the most profit and have the most productivity in terms of growing vegetables, this is well informed by data. There's clear methods and procedures to maximize growth and healthy outcomes. It's based on exposure to sun, exposure to water, certain types of nutrients in the life cycle. It considers epigenetic effects, which I'll tell you about shortly. And there's a sort of a clear science of thriving. And this is driven by motivation for profit and capital. And of course, it's also much easier to figure this stuff out when you're growing plants as opposed to humans. We might need to mute some people in the background there. So one principle that is sort of well established in pediatric health is something called the developmental origins of health and disease, or DOHAD. This was first sort of is discussed, or a theoretical model that was put forth, I think, in the 70s. And it really posits that there are important individual differences in disease susceptibility, and that's correlated with differences in immune development. Remember, I was just talking about how the immune system is the consummate biological system that is informed by the environment and experience. So DOHAD posits that adult diseases can be traced back to childhood, even perinatal or in utero exposures. And it's the idea that responses to environmental exposures confer short-term survival, but also has lasting implications for immune development and later health. And the immune system is the consummate developmental origins of health and disease organ, including a parallel that early life programming directs later immune health through epigenetic mechanisms. So I'm putting this forth as a model to think about how a parallel exists in brain development and how early childhood exposures also confer risk for later life adult mental disorders. So a model system for this process is the development of the immune system. The immune system is an organ that specializes in responding to environmental exposures. It is designed to differentiate things that are inert from things that are toxic. You don't want to respond to things that are innocuous, because then you have an inflammatory response that is not healthy. We see that in inflammatory disorders, autoimmune diseases. The immune system has the capacity to perform this task, and it develops in response to exposures in early childhood. This is the reason why we immunize children in early childhood, because their immune system responds and then confers protection for the rest of their life. It's an ideal model for how a biological system is informed by experience, both to the advantage and to the detriment for later health. And I'm arguing the same thing is occurring with the brain. So here's just one example of a developmental origins of health and disease model. So babies who are born in the fall and the winter, who stay inside more because it's cold in the Northern Hemisphere, and they are exposed to less light, they are more vulnerable to develop eczema, because they have poor vitamin D synthesis, and eczema, with the eczema, they have excessive antigen exposure. This develops sensitization and food allergies and more allergic reactions. So this is just a model of how the early environment is priming the system in one way or another to develop a disorder, or when this isn't the case, to protect from a disorder. Another way, and another mechanism by which early experience impacts the brain in particular, or other systems or other biological systems, is through epigenetics. So epigenetics is how we now understand how nature and nurture come together. Epigenetics is essentially a mechanism by which experiences in the environment change gene expression and change biological systems. And in this case, we're looking at brain. So these were some landmark studies that were done in the 80s originally. And the model here is to look at rat pups, and to have rat pup mothers who are conceptualized as being very, very nurturing. They engage in a lot of licking and grooming behavior with their baby pups. What was discovered is that that behavior, the act of licking and grooming, actually causes a change in the structure of the DNA, which causes RNA to then express certain types of glucocorticoid receptors that go to the brains of the baby mice, increase the development of the hippocampus, which is a region of the brain that is important for adaptive stress reactivity. So those hippocampi become bigger, they have more glucocorticoid receptors. Those baby rat pups become more regulated emotionally, and they engage in high licking and grooming behavior themselves. So this is just sort of a model that is well mapped out, that sort of shows how early nurturance actually changes gene expression and brain to change the behavior of the offspring. Now, of course, this is an observation that psychiatrists have known about. The behavioral relationships we've known about for years and years and years, and have made these observations, but this just gives us sort of the physiologic mechanism by which this takes place. So just a parallel to this that we stumbled upon in our study sample that I told you about, that we've been following for about 17 years. In this study sample, we observed these preschool children with their primary caregivers in the lab, and we exposed them to mildly stressful conditions. So we were sort of stressing the caregiver. In particular, caregiver and child came into the lab. They underwent a procedure where the child is given an attractively wrapped gift sitting in front of them, but they're not able to open that gift until the caregiver caregiver completes a pile of paperwork. And the caregiver, of course, the child becomes very, very agitated, wants to open the gift, they become restless, and the caregiver has to respond to the child and keep them from opening the gift while they're performing this other task. And using that, we rate caregiver sensitivity. And what we found is that caregivers who were more sensitive in this task, who were more nurturing, gave more positive reassurance, had children who had larger hippocampi when we scanned their brains at school age, as opposed to the caregivers who were more irritable, more harsh, or more punitive. So that's kind of a parallel to what I just showed you in the animal model. We don't know whether it's an epigenetic mechanism, but we assume that it is. Similarly, in the same study sample, I think I already mentioned that we found very, very strong relationships to socioeconomic status, which we've conceptualized as an income to needs ratio, or poverty status. This is how the poverty line is established. The poverty line is defined by the federal government as a certain income by family size. And when you're below a certain level, you are officially living in poverty. People who are even at 2 times that level can effectively be living in poverty. But we know from our data, and now many other data sets showing that exposure to poverty early in life has a very powerful negative impact on brain development. What this shows us is that this effect is mediated by caregiver support. So what we also know is that caregivers who are facing poverty, who are sometimes needing to have multiple jobs, juggling multiple balls, have less time to parent, have less capacity because of the demands on them to be supportive parents. So we know there's a relationship between low income to needs and your capacity to be a supportive parent. And we also know that the effect of income to needs, the negative effect of income to needs on the hippocampus is mediated through parenting. So that's another important piece of information, and suggests that targeting parenting for children living in poverty is very, very important. Now, another thing, just to sort of further underscore the power of nurturing parenting, our newer data in the field of health psychology that show us that parental warmth also has an effect on the immune system. So what we know is that children who are reared without a warm primary caregiver and children who are reared and exposed to adverse conditions have an abnormal, unhealthy immune response that we call the pro-inflammatory phenotype. This pro-inflammatory phenotype is essentially sort of an upregulation of your immune response. Your immune response is on alert to look for toxins in the environment. And what we know is this is the type of thing that gives rise to autoimmune diseases. So it's not healthy to have this pro-inflammatory phenotype. It's sort of like having a stress response or chronically high cortisol levels. What we know is that when those children have high maternal warmth, that decreases the pro-inflammatory phenotype. So that's another really, really important caveat of the importance of maternal factors and maternal nurturance, not only on brain, but also on the immune system. Okay, so there we go. So what we've sort of argued in a recent paper that we published in Trends in Neuroscience is the idea that these principles can be used to think about how to develop a formula to optimize brain development in humans. The idea is that animal studies need to be done to figure out how to optimize these systems, to sort of unpack when are sensitive periods for specific skills and abilities, because they may be different for different skills and abilities. They may have regional specificity, and that we can apply these things to humans early in development in a very proactive way, particularly for children facing adversity, but frankly, I think for children overall. So it's developing a formula for optimal child development. So what we're saying is developmental data is needed to inform how the effects of the psychosocial environment, broadly defined, impact brain and behavioral development. This will create an empirically informed guideline for when it's most important to protect children from adversity. Obviously, we always want to protect children from adversity, but let's say the brain is much more sensitive to adversity during the period between 12 and 24 months. That would be very, very important information to know that could inform public policy when we have a society that is hesitant to just broadly and robustly invest in early childhood development. And then to look at the regional specificity of these things and the skill specificity of these things. So this is all contingent on this idea of the sensitive period or the critical period. And we know a lot about sensitive and critical periods in certain arenas, like, for example, in visual systems. We know a lot about critical periods. We know that there are critical periods where if the brain isn't exposed to light during those certain periods, you'll have blindness, and that will be irreversible. And this has informed pediatric practice for when strabismus and problems like that are corrected. So a sensitive period is a period of heightened plasticity when the effects of experience on brain architecture are enhanced. They're neurobiological processes that involve the timing and balance of things at the molecular, cellular, and circuit level, and that they control the opening and closing of sensitive periods. There's new and emerging data on how to actually open sensitive periods later in life that is very promising, suggesting maybe some of our psychotropic drugs might actually open sensitive periods. So this could have some impact on older children and adults as well. So key considerations when we think about the public health applications of sensitive periods is, one question is there regional brain specificity? Are certain brain areas more sensitive to the environment than others at certain times? Does it depend on the nature of the experience? There's actually quite a bit of data now that suggests the nature of the experience is important. Whether you're exposed to deprivation or whether you're exposed to threat has a very, very different impact on how your brain is developing. Whether you have disrupted caregiving early in life or you don't has a very, very different impact on how your brain is developing, as well as the timing, of course, as I just elucidated. So animal models can really, really help us understand the causal relationships here. And there have been a lot of really interesting animal studies and animal paradigms. Things like if you look at developing rat pups or mice and you separate the mother from the pups, this has a very important negative impact on the brain development of the rat pups. You can also simulate poverty in these animal models by having a resource scarce environment. So in other words, you have a rat in cages who are rearing baby rat pups, and there isn't a lot of food or bedding available. Those mother rats then scurry frenetically around the cage, looking for food, looking for resources, and therefore do not have enough time to lick, snuggle, and nurture their baby rat pups, and those baby rat pups then have negative neurodevelopmental trajectories. So these are ways in which we can model these things in animals and then look at causal relationships of experiential factors and brain. And there are sort of regional differences here. Key areas that we look at are the hippocampus, which I've just shown you a lot of data on the hippocampus. That's a region that is very stress responsive. It's where a lot of glucocorticoid receptors are. It's very, very important for emotion regulation. It's also important in memory. Amygdala is a key region that's sort of linked to emotion reactivity. And the striatum and the prefrontal cortex are two areas that are sort of cortical areas that have top-down control, and they're engaged in response to pleasure, they're in response to reward and regulation and sort of cognitive control of emotions. So we know all these key brain areas involved in emotion circuitry are very, very much impacted by early environmental and experiential factors. So I want to say a little bit about the role of microglia in neurodevelopmental disorders. And this is where we tie together the immune system and the brain. So the microglia is a brain cell that is essentially the immune cell of the brain. It's a macrophage that exists in the brain. It's known to be highly responsible to environmental influences, as are all immune cells. It has 3 phases of growth. There's sex differences in the response of the macrophage. The macrophage is an important link between the immune system in the brain and the microbiome in the brain. And the macrophage is very much involved in pruning. Pruning is a process by which early in life, the brain sort of becomes more efficient. There's a loss of brain cells. The brain gets sculpted and pruned down to a much more efficient sort of way of processing information. And this is an important thing that is influenced by environment and by sensitive periods. And the macrophage is a key cell in that way. So the macrophage also influences the microbiome, and the immune system is influenced by it. So other things to think about are the fact that exposed to toxins, exposed to exposure to things like bacteria and viruses and things like that are also may have an impact on brain, may have an impact on the microbiome, et cetera. I mentioned also that sleep is another area that we can target when we think about early experience in brain development. And we know that sleep plays a very, very important role in neurodevelopment, particularly in neurocleansing, and that this is also tremendously important during the first 5 years of life. So this is something that we need to sort of learn more about and could inform more about how we should have children sleeping during the first 5 years of life. So if we think about optimizing immune development, I mean, a question is, when is it best to expose children to different doses of pathogens? This is similar to the idea of how to time immunizations. Little is known about the development of the immune system and how it interacts with other environmental exposures. I think there's a lot of emerging data suggesting sleep impacts the immune system, physical activity impacts the immune system, and all of these things impact brain. So this is what I'm trying to sort of get at, that all of these factors that are changeable and modifiable could be harnessed to sort of develop early health promotion. Let's see, I think I talked a little bit about that. So when we think about the microbiome, this is an area that has become of great interest, just thinking about general health. So we know that the microbiome is essentially a living system of microbes that exists within our guts, that grows in early childhood. It is very responsive to environmental exposures, not just diet, but stress and adversity. People use probiotics to change the composition of the microbiome. There are ways in which we could optimize the microbiome. We're having increasing appreciation, obviously, for the effects of drugs that are used to treat infections, antibiotics that kill the microbiome, and how this could impact children early in life. So again, this is another avenue by which we can impact health and impact brain development by modifying the environment. So essentially what we're talking about is thinking about how to grow healthy children by enhancing their exposure to nurturing caregiving, avoiding separation from caregivers at certain key times, educating caregivers on how to behave in a more nurturing fashion, thinking more specifically about when there are sensitive periods, thinking about when there are key periods when protection from adversity is most critical, and then thinking about when to expose children to things like language learning, social contact, exposure to peers, sleep, etc. So this is just a little bit of data to show you some of the things we've learned about how experience changes brain development in a regionally specific fashion. So looking at our 17-year study with 5 waves of brain scans and sort of following children who are developing and tracking their environmental exposures, we know that there are differences in different regions in terms of their response to maternal support and response to adversity. So this is the pattern that the hippocampus follows in terms of differences in exposure to adversity at certain levels of maternal support. These are the 2 key things that we've shown impact brain. So here's a hippocampal pattern. Here's an amygdala pattern that you see is very different. So again, a lot of important regional specificity to think about. The hippocampus, remember I talked about, is an important emotion regulatory region. Amygdala is more of an important emotion reactivity region. And then here is the caudate, which is a part of the striatum, also has a very different trajectory. And that is also more of a emotion regulatory sort of cognitive control region. So again, just showing that there is regional specificity that needs to be considered. So this is just sort of a big picture model, sort of if we think about developing children, and we think about some children are growing up in a context of general adversity. They're living in poverty, they're living in neighborhoods where there's high levels of violence and crime. And in those circumstances, even in those adverse circumstances, there are ways in which we can target the parent-child caregiver and enhance parental sensitivity, predictability, and nurturance. There are ways in which we can enhance stimulation in preschool settings. We can inform what sleep should be like or diet. Or we can protect children during certain very specific sensitive periods from adversity. So all children are going to be exposed to adversity and negative influences invariably, but in adverse circumstances, these types of protections are even more important. These types of stimulations are even more important. But we can also think about children being reared in more optimal conditions and how these principles would be very important for those children as well to optimize their neurodevelopment. So this is just, when I don't know if you all remember when the infant stim mobile first became very, very popular, and everybody bought one because we thought, well, if we can make our babies smarter, this is what we want to do. I mean, essentially what I'm saying is that things in the environment can be manipulated to enhance neurodevelopment and make babies smarter and more emotionally adaptive and more resilient to risk for psychopathology. So if we ask the question, how early does adversity begin to impact the developing brain? So until very recently, the earliest findings have been as early as about 6 months of age, or as early as about 5 weeks, we see effects of the environment on brain change. We've recently embarked on a large-scale study that we call the ELAB study, Early Life Adversity, Early Life Adversity and brain change, basically. And what this study is doing is we have a sample of mothers, about 400 mothers that we followed very closely during pregnancy. During pregnancy, we measured maternal depression, we measured maternal diet, we measured maternal stress. We actually took blood from the mothers, so we have measures of maternal inflammation. We get measures of the maternal microbiome at birth. And then we're following the offspring. Actually, right now we're following them birth to 3 years of age. What we did is we did brain scanning on about 400 of the babies at birth. And we are also looking at their developing gut microbiome. We've been looking at the development of their immune system, and we've been bringing into the lab, looking at their behavior and looking at their interactions with their mothers. And what we found so far are very, very surprising and powerful influences of the psychosocial environment that the pregnant mother is experiencing on infant brain at birth. So essentially, we know from our theoretical models that as stress experienced during pregnancy can get transmitted to the fetus by a variety of mechanisms. We know that when women in pregnancy are exposed to stress, they have elevated cytokines, which is sort of a signal that the immune system is on alert. We know that they have high levels of cortisol. We know that they have high catecholamines. We know it influences the maternal microbiome. And all of these things cross the placenta and into the baby and may influence the brain maturation of the baby. So I'm actually talking here about literally psychosocial influences. Not even these things can have an effect above and beyond the mother's health, above and beyond the mother's diet. All of those things are important, but psychosocial influences in pregnancy. Oops. Okay, there we go. I'm just filling in the slide here. And this is influenced by poverty, maternal education, obviously things like lack of medical care, trauma, et cetera. All of these things are influencing the mother and effectively influencing the developing fetus. And what we know is that pregnancy is a sensitive period for neural growth. We know that neural growth is just proceeding at a very, very rapid rate in utero and in the first year of life, which gives us this incredible opportunity to influence neurodevelopment during pregnancy and the first year of life. And basically what we found thus far in our study that I just showed you is that mothers who have low socioeconomic advantage, in other words, mothers who are exposed to poverty, who experience high levels of psychosocial stress, have babies that have smaller brain volumes at birth. So this is before they're even exposed to the environment. And this finding holds up even when we control for the health of the mother, we control for the diet of the mother, and we control for the medical status of the mother. So essentially, this is showing that adversity experienced during a pregnancy is powerfully influencing the fetal developing brain, and this is evident at birth. These findings are pretty global, and they are also not trimester specific. So this leads us to sort of understand how important the psychosocial environment of the pregnant woman is. We also found very, very similar differences with cortical folding, which is another measure of infant brain development, all very, very important for later neurodevelopmental outcomes. And then another really surprising finding, just underscoring the importance of sleep, is that we found that one of the ways that maternal disadvantage appears to be influencing infant brain is actually through maternal sleep. So when we look at our mothers, the other thing I neglected to mention is that we are tracking sleep during pregnancy in these pregnant women. These women are wearing actigraph watches. We know a lot about how they're sleeping, and we know that women who have chronodisruption, who do not have regular sleep patterns, have babies who have smaller cortical gray matter volume. And so basically maternal disadvantage is acting through disruption in sleep. Again, underscoring the importance of these experiential factors, even in utero, on infant brain development. So these, just to summarize, these are the emergent findings in our ELAB study. All of these things are under review in the literature right now. None are yet published, but I hope they'll be published soon. We know that poverty has a very powerful impact on structural brain development at birth. These effects are global and they're not regionally specific. We also know that it impacts the tracks as well. I didn't show you this data, but we have data that shows it impacts the connection, the structural tracks between different regions, which implies that there's going to be changes in function. And those changes are more specific. We also know that maternal sleep and chronodisruption, that is higher sleep onset variability, women who don't go to sleep at the same time every night. This is like the worst case of this is a shift worker or a medical resident, for example. When this is happening during pregnancy, this has a very important impact on neonatal brain outcomes. And whenever the trainees in residency hear this finding, I get a lot of reactions and questions, as is very legitimate. So the impact of these findings on brain change, if we sort of start to focus, why is this important for psychiatric disorders? We know that we can start to detect meaningful psychiatric disorders, even categorically defined as early as 3 years of age. We know that these neurodevelopmental changes are markers of early risk for these psychiatric disorders. So looking at these early neurodevelopmental changes may give us better tools for the earliest possible detection of risk during this neuroplastic period for early preventive interventions. And essentially, we know that emotion dysregulation and its neural correlates are very, very detectable early in life, as early as infancy, and that these could become markers that we begin to target, that we are now sort of ignoring in primary care settings. Now, some of the unique challenges to the earliest possible detection of mental disorders is that there's a lot of high normative variation in infant and early childhood behavior. There are a lot of dysregulation, irritability, temper tantrums are normative, and we don't have enough data that really maps the difference between these normative variations and what crosses the clinical threshold. And we also have to consider sensitive periods in in that formula as well. So these are challenges that lay ahead for us. But sort of, if we think about the importance of these findings to adult psychiatric disorders and even to adult outcomes, this is an article that was published in, I think it was JAMA Psychiatry, from the Dunedin study, which is a study that is an epidemiologic study that looked very, very intensely at over 300 kids starting at age 3 and now into adulthood. And these investigators essentially showed that emotion dysregulation early in childhood was a predictor of not only adult psychiatric disorders, but also of adult medical disorders, and even things like, later dementia and other types of chronic diseases. So this just sort of underscores how important this early neurodevelopmental framework is more broadly to our field and to health outcomes in general. So what I'm arguing is that these principles should be used and can be used to sort of inform precision health promotion or health enhancement. This is something we've paid surprisingly little attention to, and our understanding of these risk trajectories and how they're informed by sensitive periods will give us the roadmap to optimize neurodevelopment. And we need this data because right now we're sort of shooting from the hip. So we're sort of arguing for precision developmental science that focuses on optimizing health and development, to educate primary caregivers, to utilize socio-emotional, immune, and microbiome growth charts or risk calculators to enhance prevention and general health. And just with that, I want to just give a lot of credit to my key collaborator, Deanna Barch, who's the neuroscientist on our team, and the group of really excellent young investigators who all have contributed to this work. And with that, I'm going to open up for questions that you guys might have.
That was lovely, Dr. Luby. Thank you so much.
Thank you.
Incredible overview and precision at the same time. Maybe people who have questions or comments could begin putting them in the chat, and then we could get some type of order to comments or questions. And maybe to start us off, I had a quick question. So this is very powerful work that you and others have been doing. Is there any similar research on overcoming these deficits in kids based on the risk factors? Interventions that have happened and been able to reverse some of the brain changes that may have occurred already?
Yeah, that's a great question. I mean, there actually are several really, really promising early interventions that have been tested, that have been empirically proven, some of them sort of home visitation models, some of them video-based parent-child interaction coaching models. We actually have been working on an intervention for early childhood depression that we have now modified into a more global, broad preventive intervention. So there are, I'd say, a handful, about a half a dozen that have been developed that are relatively short, are fairly easy to learn. So I do think this is a very, very promising area.
Thanks. So there's a question from Dr. Sugarman, who's curious about whether corporate America has attempted to bias or impact your work or findings.
I don't think so. Can you say anything more specific?
Peter, anything to add from that?
Oh, just, I was thinking back to when I was raising my children, and people were using baby Einstein or music in order to enhance the development of our children. So I was imagining that this work would lead to all sorts of corporate products, but I was curious whether that was happening.
Oh, it could be, there could be a way that that could happen and could be very positive. Unfortunately, I haven't seen any dollars from corporate interests yet. I mean, if they could certainly fund studies like this that could be productive for all. Thus far, all of the funding that we've had has come from the federal government, and any serious studies that have been done on this have all been federally funded. So I don't know that corporations have gotten interested in that yet. But I could see that in the foreseeable future, and hopefully we can keep it scientifically, under control and honest.
Anybody else have questions or comments?
So I just have a question. My name is Sadiq Naveed. I'm actually doing my infant fellowship from UMass Boston. How do you conceptualize the role of fathers compared to mothers in early caregiving? Do you see any different response in data, or how do you see that?
I always get this question, and I always say to myself, I need to fortify my lecture. I mean, really, the data that I've shown you, it pertains to caregivers, broadly conceptualized. That could be fathers, it could be mothers, it could be grandmothers. I mean, in the study sample that we have, something like 96% of primary caregivers were mothers, but we've done interventions with primary caregivers that were fathers, that were foster parents, that were grandmothers. So I don't think that there's a primary caregiver is a primary caregiver. I don't think it really matters exactly who they are.
Great question, Sadiq. Evan?
Yeah, Dr. Luby, thank you so much for an incredible lecture, because I think in our field, we've lived so long with theoretical models and inferences and suppositions, and really this longitudinal study is quite profound. And I guess I have a even more special interest since my daughter just had a child, and it's nice to validate at least some of what she's doing. But more to the point, when we review what's going on in our government, and more specifically the latest proposal that has been denied regarding family and medical leave, and you indicating that this is government-funded research, has there been any conversation with, dare I say, the Congress, to inform them regarding this part of an otherwise very large $1.75 trillion proposal? And do you think that they even have the capacity to hear the consequence of not just this work, but also the work on education and ultimately our workforce one generation from now?
Yeah, that's such a great question, and something I've been really thinking about and feeling very preoccupied with as I watch this stimulus package struggle. The connection between the scientific research and the public policy seems to be a very frayed and mysterious one. As we know, I mean, we're struggling in our society with the question of acceptance of scientific fact writ large, more broadly. There's always been a disconnect between how science impacts public policy. Of course, we have a much better opportunity to do that with the current administration. I mean, but yes, it's very, very disheartening to see the ways in which, some of these really, really important policies have been shaved off of the current stimulus package. And I think it really has more to do with short-term gain versus long-term gain, of course. But I've never had the opportunity to be involved in any of those discussions where the federal government is being informed and influenced. And I think that's a sort of mysterious process where it seems like you have to have a member of Congress who has a particular interest in one. They've heard a lot about autism, for example, when because these are really, really salient facts that should be influencing these policies, but yet there just seems to be a gap that, I really hope it can be bridged in the next several years. And obviously that does depend very much on the politicians, their willingness to listen to scientists, et cetera.
Thank you very much.
There's another comment in the chat with a question. Dr. Luby, you spoke about some of the mediators of child brain development. Has the Biden child tax credit provided a natural experiment? And do we see any data from it yet? Is anyone tracking the epigenetic effects? And if not, or you aren't sure, for what sensitive period would you anticipate it would have the greatest effect?
That's an excellent question. And yes, so the Biden tax credit, which moves like 50% of children out of poverty, is a beautiful natural experiment. And we hope that we are actually collecting data on that natural experiment in our ELAB study. So this policy has gone into effect as we're following that study sample of the offspring of the pregnant women. Most of those kids are now between about 2 and 3. And we're conducting brain scans at age 2. We're continuing to collect data on them at age 3. So we are thinking that this will be a natural experiment, and we hope to explore whether there's a significant impact on these kids in our study sample.
And another question, is there a trimester of pregnancy during which maternal well-being has a particularly large effect on children?
We haven't found it in our study. That doesn't mean it doesn't exist. But we haven't yet found it. It could be our sample size isn't big enough, but thus far, we don't find any trimester effects.
Okay, a question from Dr. Adesso. How do you account for the children who have experienced adverse life events and or come from lower income homes who have thrived?
Absolutely. That's I think very, very consistent with a lot of things that I've just presented. What I'm sort of saying is that children who are experiencing adversity, who have a nurturing caregiver, can thrive, because I think a lot of the detrimental effects of adversity do get sort of conferred through the caregiver. So it is possible to be experiencing poverty, adversity, and have a supportive caregiver and do incredibly well.
Dr. Naveed, you have your hand up from a reaction. Is that another question you have?
No, I don't have any other questions. Thank you.
Okay. Another question came up. How do variations in hippocampal and amygdala volumes correlate to effects or development of mental illness? Is lower volume associated with lower functioning or excessive pruning?
Yes and yes. And in our study sample, lower volume is associated with poorer function. It's true in several other studies as well. We don't know whether that has to do with pruning or not. We assume it probably does, but we would need studies where we have brain scans much, much earlier in development. We're now, of course, collecting that data, but thus far that has not existed. So we also know changes in amygdala size correlate with changes in function, with changes in anxiety levels and increased reactivity.
Okay. Any other final
Uh,
comment to
Yes,
question?
I have a question.
Thanks.
Have you detected any differences between children raised in the home by parents versus children in their earliest years cared outside of the home? You know, the kids who go into daycare very early.
We haven't looked at that, although there is a very, very big study that was funded by the NICHD on the effects of daycare on child development, that was sort of looking at it through an attachment lens. And they kind of concluded from that study that daycare before 6 months of age, it tends to result in poorer outcomes. After 6 months of age, the outcomes are good, and it very much depends on the quality of the daycare.
Okay, thanks, Hank. Anybody else? No other questions, comments? All right. Dr. Luby, heartfelt thank you again for your persistence, and it certainly paid off. We're very happy we finally were able to complete this with you.
Thank you so much. I really enjoyed talking with you.